This video introduces fast and efficient methods for the synthesis of magnetic nanoparticles and the use of magnetometry to follow their transformation in the biological environment.
Magnetic nanoparticles have attracted increased attention for biomedical applications, and it is important to understand their fate once internalized in cells. This is achieved here by measuring the cellular magnetism.
For both magnetic nanoparticle synthesis and measurements, results could be affected by contamination. Use well-cleaned chemical vials and make sure that the magnetometer equipment is free of contamination. The magnetometry samples holder is usually used for powder samples. We show how to adapt the use of the holder to liquid or biological samples.
In a 30 milliliter monowave glass vial, dissolve 400 milligrams of iron 3 acetylacetonate in 10 milliliters of benzyl alcohol. Using a microwave reactor, heat the suspension from 25 degrees Celsius to 250 degrees Celsius at a rate of 11.25 degrees Celsius per minute, and maintain it at 250 degrees Celsius. To separate the nanoparticles, apply a neodymium magnet for 30 minutes. To pellet the nanoparticles, perform a series of washes using 10 milliliters of the designated wash fluid and applying a neodymium magnet for 5 minutes. Remove the filtrate and add 12 milliliters of 10 to the -2 molar hydrochloric acid. Centrifuge the suspension in a 100 kilodalton filter at 2,200 times G for 10 minutes. Then resuspend the